Continuing from last video, this is the tRNA, which is exiting the ribosome from the E site. There's an exit site, and then you see one is entering into the A site, which is another tRNA with the specific anticodon sequence for the next codon in the mRNA sequence, and then you see this has the polypeptide chain in the P site. Lastly is termination. So after that goes around in a cycle in order for a long time, something called a release factor is going to bind in the it's going to bind in the P site of the A site of the ribosome. So once it binds in the A site of the ribosome, this is going to initiate for the ribosome to dismantle. So the messenger mRNA is going to dismantle from the ribosome, large and small subunit of the ribosome, and then you'll have a free polypeptide chain in the cytosol. So what actually happens here is that it's going to go through either its primary or it go through primary to quaternary structure to become a protein. So another few facts you should know is that first in mRNA it takes well, you should know that all about the anticodons and such, but for once it's released, it becomes a protein from the primary, quaternary, and tertiary and quaternary structures. And then after that, what happens is, depending on where the protein was synthesized, it depends on what actually it's going to be used for. So if it was, if the ribosome that synthesized it was attached to the ER, then the rough ER, then actually it will be used outside the cell or in the plasma membrane, because the ERs are part of the intermembrane system, and it will transport these proteins to the cell wall, into the plasma membrane when by the Golgi apparatus, transport vesicles. And then, but if it was synthesized in a ribosome in the cytosol, then it will actually be used within the cell. So the protein will be used within the cell because the cytosol the protein synthesized in the cytosol. So it will be used somewhere in the cell. Therefore, I actually believe that's it. That goes through, first it goes through the DNA structure. We went through the DNA structure, what deoxyribonucleic acid stands for, its structure, what it has in it, transcription, the 3 steps of it, and then also translation, the 3 steps of that, and what it actually means for both, and then the aftermath of what happens to those proteins after, what they're going to be used for. So again, thank you for watching. Have a nice evening.